Well, welcome everybody to this inaugural University of Toronto LMP Alumni Career Panel Night. I'm Gino Somers. I'm Division Head of Pathology at the Hospital for Sick Children and Professor of Pathology in LMP. I'll be hosting this evening, and joining me for the Q&A session is Maryam Khalil. Maryam is a U of T double major and a PhD student in Dr. Ming Tsao's lab at Princess Margaret Cancer Centre. And she's also co-president of CLAMPS, our graduate student union. So these alumni events came together really as an effort to improve the connections between students and faculty, both past and present. And that was a desire that was expressed at several levels throughout LMP, through surveys and town halls. And we've, as in Dr. Michael Polanin, who's our Vice Chair of Innovation, and Jenny Bozek, who's our Communications and Outreach Officer, we've put together 3 of these panels for the year. This is our first panel, and it's focused on careers as clinicians and scientists. Our second panel will be sometime in the late spring, and will be focused on alternative careers after leaving LMP, such as law, consulting, and corporations. But tonight, we are bringing together 3 successful alumni from LMP who've carved out careers as both clinicians and scientists. And I'd like to introduce them now as one, and then they will all speak. And at the end of the third speaker, we'll have the Q&A session. So our first speaker, our first panelist, is our LMP Departmental Chair and Professor Dr. Rita Candel. Rita completed her medical school and residency in anatomical pathology at the University of Toronto. And she is a clinician investigator. And in addition to her roles in LMP, she is also the Chief of Pathology at Mount Sinai Health System and Women's College Hospital. She's cross-appointed to the Department of Surgery and is a member of the Institute of Biomedical Engineering at the University of Toronto. She's also a fellow of the Canadian Academy of Health Sciences. Her clinical expertise is in the pathologic diagnosis of soft tissue and bone tumors, and her research interest is in the bioengineering of tissues for articular cartilage and intervertebral disc repair and replacement. Her lab is composed of a multidisciplinary group of investigators, including engineers, biologists, stem cell biologists, and clinicians who work together to develop novel treatments for cartilaginous diseases. She has published over 240 papers and multiple book chapters, and has trained numerous undergraduate and graduate students and postdoctoral fellows. Our second panelist is Dr. Phedias Diamandis. Phidias is an alumnus of the U of T MD PhD program and completed his neuropathology residency in LMP. His graduate work focused on designing high-throughput screening platforms to chemically profile neural precursors. And this work resulted in the identification of novel regulators of neural and cancer stem cell function. Following the completion of his work in 2016, Phidias was appointed as a scientist at the Princess Margaret Cancer Centre and as a neuropathologist at the University Health Network. His research now focuses on chemical biology, deep learning, and mass spectrometry-based proteomics to resolve phenotype-level heterogeneity in different brain and glioblastoma niches. And our third panelist is Dr. Susan Doan. Susan completed her medical training at Cambridge University in the UK and then moved to Canada, where she completed a residency in anatomical pathology at the University of Calgary. She then moved to Toronto for her fellowship training, where she also completed a PhD at Mount Sinai Hospital and an MBA at the University of Toronto. Since then, she's been a pathologist at the University Health Network and a researcher at Princess Margaret Cancer Centre. She's currently Associate Professor in LMP with a cross appointment to the Department of Medical Biophysics, and she's also the director of the Anatomical Pathology Residency Training Program. Her research is predominantly translational, and Dr. Doan is focused on breast cancer intratumoral heterogeneity, circulating tumor cell markers, and early events in invasion and metastases. But she also has quite a rich collaborative research activity, including molecular epidemiology, artificial intelligence, and novel radiopharmaceutical agents, all focused on breast cancer. So before I hand over to our first speaker, Dr. Rita Candel, I just want to say a couple of housekeeping announcements. Firstly, this event will be recorded. And secondly, if you do have questions, please enter them in the Q&A function or in the chat function. Either way, we'll be able to get to them and hopefully get through most of the questions in our Q&A function, in our Q&A session at the end of the third speaker's presentation. So without further ado, I'm going to hand over to our first panelist, Dr. Rita Candel. Rita.
Thank you, Gino, for the kind introduction and the invitation to speak today. I must admit, I don't think my story is special, but I'm glad to share it and hope you find something of value in it. I'm going to start by sharing my screen. So I'm going to confess up front that I never had a career plan. I just did what made me happy. So here goes my journey as a clinician investigator. And just so you know, I feel like I'm still traveling. I'd like to start with my take-home message. And this message is that it's important to be open to new possibilities. If I had not had my antennae up to identify these new opportunities, I believe my career wouldn't have unfolded as it has. I grew up in a immigrant family who struggled to make ends meet. We didn't have much expendable money for things other than food and shelter, so I started working at the age of 11. As high school ended, I learned that my parents didn't believe in higher education. It turned out to be fortuitous that I was working, as it provided me the freedom to pursue my goals without their approval. In retrospect, I think it also allowed me to see early on that I was not going to allow anyone to tell me what I could or could not do. As a result of my precarious financial position, I was a Toronto lifer, born, bred, and educated in Toronto. I did my undergraduate arts and science studies and medical school at U of T. I had decided in high school that I wanted to become a doctor. I was attracted to medicine for a number of reasons, but one of them was that I saw medicine as my way to help others. I was so thankful that Canada had accepted my parents and allowed me a safe harbor and a place to launch my life. My choice to specialize in pathology was quite fortuitous. When I was doing an elective in fourth year medical school, the medical resident, who I think was really quite tired of me, asked me to follow up on a patient's biopsy. A biopsy is when we sample a piece of tissue from a tumor, for example, to make a diagnosis. So I went to the pathology department, and there I encountered two pathologists, who I subsequently found out were internationally renowned in their field. They were arguing about a diagnosis and not about whose patient would get the hospital bed. I was taken by the fact that pathology was the best of medicine without any of its frustrations. I was so intrigued by this that I stayed in the pathology department and didn't go back to the clinic for the rest of my time. This experience exposed me to the thoughtfulness and rigor of the field and the role the pathologist played in ensuring the patient received the right treatment. About 80% of medical decisions are based on results from the lab medicine program. That is an awesome responsibility. The integral role of all disciplines of lab medicine and patient care has only become more apparent over time with the advent of genomics and precision medicine. This chance encounter taught me that career decisions are often influenced by the people you meet. A good reason why it's important to take advantage of all opportunities. I was accepted into the pathology residency training program at U of T, as Gino mentioned. And during my residency, I discovered cartilage, a tissue that you can see here on the screen that covers the ends of the bones. Cartilage allows us to perform the acts of daily living and survives even though it doesn't have a vascular supply. How remarkable is that? However, the consequence of this is that when it's damaged, it can't repair by the usual fibrous process that occurs in other organs. I realized I wanted to spend some time to learn about this tissue. So I spoke to Ken Pritzker, a musculoskeletal pathologist in Toronto at the time, and told him I was going to apply for a CIHR fellowship so I could do a year of research during my residency training, and I wanted him to be my supervisor. There was no clinical investigator program in those days, and if I wanted to immerse myself in research, I would have to find the funds to do so. When I spoke to the department chair at the time, he was quite supportive of my desire to take a year off clinical training. But he did try to convince me to do cancer research, his area of interest. I was honored that he wanted me to change my focus to something he was passionate about. He was a preeminent researcher, and I interpreted this as a sign that I must have some potential if he wanted me to do what he was doing. But I knew what I really wanted to do was cartilage research. I did get the fellowship, and after that year, I realized that I would need to combine research with the practice of medicine if I was going to contribute in any way to advancing knowledge in the field in a way that would give me satisfaction. So at the end of my clinical training, I applied for another fellowship, this time to do training in Boston. I had to interview for that fellowship, and I was shocked when one of the committee members started lecturing me on my audacity of wanting to take a spot away from a male whose whole commitment was going to be research. I, on the other hand, he said, would be distracted by raising a family and probably would never end up doing research. I fought back, but I could see the others on the committee wondering if I was going to be a difficult person. And when I was awarded that fellowship, I could not help thinking I got it only because they were afraid I was going to file a complaint. Nevertheless, the fellowship allowed me to immerse myself in research for 3 years. I did not do any clinical work during that time. And I loved that experience, the time I spent at Tufts and Harvard as I trained to be a researcher. But I was troubled. I understood why we grew cells as a single layer in culture. But I knew from my pathology training, this did not make sense. When I looked down the microscope, and you can see on the screen what I can see down the microscope, I saw cartilage tissue in which the cells are surrounded by a lot of this blue extracellular matrix. This had to influence cell function in some way because the loss of matrix leads to osteoarthritis. How could studying cells in the absence of this matrix be appropriate? So when I was offered a job at Mount Sinai Hospital, I knew where I was going to focus my research. I was going to develop a way to grow cartilage tissue so I could study cells in a more physiological environment. Although this doesn't seem paradigm shifting now, you have to remember at that time, organoids didn't exist. So I was going against current practices. And it didn't escape my notice that I had thought of this project only because I combined my expertise in pathology and research. Little did I know that I was going to encounter another wall before I could start doing my research. The wall was the director of the Research Institute. He was a preeminent researcher and well-respected, and when he spoke, people listened. He told me I would never be a serious investigator as I didn't have a PhD. He was very right about the importance of a PhD, but he was wrong about me. He denied me a position and lab space in the Research Institute. And I couldn't help but think that the director was like the authorities that Peter Medawar, the Nobel laureate, referred to when he described these individuals who judged his research. It's not that they're wrong, it's just that they're so sure that they're right. This experience taught me you should always encourage people to follow their dream. You have no idea whether they have the ability to succeed or not. In the end, I was able to carve off some space in the pathology department, thanks to Ken Pritzker, and set up a small lab and started my research. It took me some time, but when I looked down the microscope and saw that I had grown cartilage in tissue culture, I realized I was seeing something that no one had ever seen before. I can't fully describe the feeling, but I can tell you the result. I became an addict to discovery. This accomplishment also helped me realize I had a more translational bent. Tissue engineering was all the craze at the time, and I could see the potential of what had been accomplished. We had created a tissue for an organ that could not repair. So it was clear I had to switch my research focus to cartilage tissue engineering. I was a physician, and I now had an opportunity to develop a novel treatment. My work was picked up by Genzyme, a very successful pharma company. They too had decided to get involved in cartilage tissue engineering, which turned out to be very fortunate for me. At the time, CHR was unwilling to fund translational research. So I struggled to convince panel reviewers that the work we were doing was significant. Not to mention, it was also a dire time at CHR when funding rates were much less than 10%. My research program survived this time of limited peer review funding because of venture capitalists and industry. And that was the next lesson I learned. You need to look at all possible funding sources, including non-traditional ones, when you need them. Around that time, I received my first CIHR grant and realized that Genzyme had only expressed interest in the work so they could put it on the shelf and keep it from getting developed. I've learned that this can be a behavior of some companies. So I took my project back. Once I had this first grant, I realized that I needed to find other scientists with whom I could work if our research was going to advance. I did tissue engineering. I was a tissue person. I needed an engineer. So I looked around and found a community who would help me push my work to the next step. These scientists made me think in different ways. For example, they pointed out generating hyaline cartilage was only part of the story. The reason cartilage interfaced with bone through a calcified zone was for mechanical reasons. Without this interface, cartilage would shear off the bone. If this interface could not be reproduced, then we had no way to repair a joint. Working with these individuals and others, I realized the importance of finding collaborators with whom you enjoy working, who are smart and interested in your project. I was able to bring together a group of scientists and engineers, and we formed the Bioengineering of Skeletal Tissues team. And that was when I realized I had a skill. I could convince diverse people to work on a problem. And together we obtained one of the first CIHR team grants. And together we solved the issue of forming this interface. My second lesson, collaboration is critical for research, but it has to be with people with whom you enjoy working. The number of individuals in the group has varied over time, but I have never done my research without collaborators. And I am so grateful to my many partners, such as Mark Grimpus, Bob Pillier, and Paul Santerre, who have taken this trip with me. As we're discussing collaboration, I'd like to mention the many students who've participated in our projects. I see them as collaborators as well, who are integral to advancing our research. They taught me so much, and I am so appreciative for the time we spent together. Students often ask me how they will know if they have what it takes to do research. My answer, the only way is to try it. Do a research course in your undergraduate studies or do a graduate degree. If you find your thoughts are always drifting back to your project, then you have your answer. If you decide that research is not for you, you still come out ahead. The rigor and critical thinking you learn during that time will be useful for almost any profession you decide to pursue. So I encourage you to give research a try. As our research program began to progress, I started thinking about expanding it to include tissue engineering a biological intervertebral disc, as it has some of the same tissues as cartilage, such as interfacing soft tissue to bone. I submitted our first grant on this topic to CHR. It got triaged, which means they thought it was so bad, the reviewers didn't even want to waste their time to discuss it. But I suspected they were wrong, and I sent exactly the same grant to the NIH. To my surprise, it ranked #1 on the review panel. I think most researchers could tell a similar story. My third lesson was don't be deterred by the lack of success of your grants or your fellowship applications. It is even truer in research than in life if you only fail when you stop trying. And as you can see in this image, we're on our way to bioengineering a disc. During this time, I was working as a pathologist at Mount Sinai. Given my research interest in cartilage, I specialized in the pathology of soft tissues and bone tumors. I was really fortunate to be part of a clinical program that was internationally renowned for treating individuals with these tumors. I was so appreciative that I could move seamlessly between these two activities, research and clinical practice, both of which I love. Interestingly, they are the antithesis of each other in so many ways. In medicine, you make decisions based on judgment and intuition and not a lot of evidence, although that's starting to change now. While research, in contrast, is all about generating sufficient evidence to allow you to make a conclusion. I must admit there were some days I found it difficult to switch my brain between these two worlds. So I tried to bring the two worlds together. I remember being invited to do a session on bone tumors. In my seminar, I intertwined the research I was doing in that area with the clinical aspects. No one asked me a single question. They were not pleased. The pathologists that had attended that session had given up their weekend to learn how to approach diagnostically difficult cases, and I hadn't provided them that information. I learned my lesson. There are times when you need to keep the two worlds separate. I'm not really sure why I decided to take on administrative roles, but when the position of medical director at Sinai Health became available, I decided to apply for it. I was really surprised that my severe case of imposter syndrome did not stop me. And when I was appointed, I was the first female department head at Sinai. And I came to realize I liked administration as it opened up a very different world. It gave me the opportunity to help people and to build things. And I was able to build a leading edge lab medicine department filled with individuals interested in academics and a desire to advance knowledge. And because of these individuals, we were able to implement a number of diagnostic firsts in Canada, such as genetic testing for precision medicine. And when the LMP chair position became available, I decided to apply. I mean, I liked administration, and I thought it would allow me to interact with even more smart people, and we have lots of them in LMP, And I could also see how this position would allow me to encourage people to work together to build new programs. And from the moment the dean offered me the chair position, my world went technicolor, and it's continued to this day. I've come to appreciate that our learners, faculty, and staff are even more remarkable than I thought. What gets accomplished in LMP, from education to developing leading edge programs to promotion to wellness activities, it's all because of their commitment to our department. No question, LMP is the place to be, to learn, to grow. I've been asked how I manage to do clinical practice, research, and administration. Each morning, I decide how I'm going to prioritize my focus that day. Some days it's clinical, some days it's research, and some days it's my family. Secondly, I have a very good ability to concentrate. Only recently did I come to know that this is described as a state of flow. Mihaly Csikszentmihalyi, who first described flow, said it was a state in which people become absorbed in such an enjoyable activity that they become blind to their external environment and unconcerned about self or passage of time. I think that describes me. However, I must admit, I would not be able to do this without the support of my family. They are amazing individuals and are the air under my wings. As I come to the end of my story, to this point in time, I hope I've convinced you about the need to be open to new possibilities. I know that doing so afforded me this wondrous career. And that's my story. Thank you very much. I'm now going to turn the Zoom table over to Phedias Diamandis so you can hear his amazing story.
Thanks, Rita. Thanks, Gino, for the invitation and introduction. That was an amazing journey, Rita, that you took us on. Mine's much shorter, but I'll try to. I tried to organize it in some sort of relevant questions that perhaps are relevant to the people in this conference. So just a little bit about me. So I'm a neuropathologist, which is a pathologist that looks at exclusively brain tissue. This includes autopsy specimens like this picture shows, but also microscopically looking at brain disorders from cancer, medical diseases, and trying to kind of reach diagnoses that will hopefully guide care or family planning. And then the other half of my job, about 75%, is being a scientist. You know, this spans a number of different areas, as Gino mentioned, including the use of computational tools to study microscopic patterns, molecular profiling, bioengineering using organoids to study neurodevelopment, and even some electrophysiological studies that we're getting into. And this is nothing, you know, I think the key thing I took from Rita that I think resonates well is I never thought I'd do any of this stuff. I never thought I'd be a neuropathologist. I thought I was going to be an anatomical pathologist, and I never expected. I knew I wanted to do research, but you know, if you're always worried about, I don't know exactly what type of researcher I want to be. I never thought I was going to do any of these things when I got my position in 2016. Every year we try to do something new to kind of continue innovating. But I never thought I was going to use computational tools, you know, growing artificial brain tissue in a lab to study neurodevelopment. And even now we're getting into, we got our first REB for real patient studying real life patients. So I never thought I'd be doing any of this stuff. So don't worry if you haven't planned it all out. So why did I choose a career in science? So it's hard to kind of pinpoint, and I think the other key point that I think Rita mentioned that I also agree with, it's every story is unique. I'll tell you mine, and retrospectively, this maybe the correct story, it may be partially incorrect. It's hard to know why. Number one, I think I had some family bias. Both my parents are PhD scientists. My dad's a pathologist, so there was some support, if not bias, towards a career in science. And even if your family's not in it, I think it's an important driver. So if you have friends that are in science, I think being able to talk about your career aspirations and passion and develop them with close people, whether they're your family or your friends, is really important. Of course, there must be an interest. I started with an interest in science, in the human body and biology, and that has evolved during my training to be very neurocentric. If you talk to any of my trainees, I'm very biased for the brain. And I think we saw Rita's very biased for the cartilage. And I think that's something that something needs to drive you. You need to have a passion for whatever you're studying, that people look at you and say, I can't understand why you're so interested in one particular organ. So I think that's a key driver. Of course, there has to be a thirst for experimentation, not just following kind of a flow chart or a set of protocols. There needs to be a desire to kind of deviate from the lines and ask questions that may not have been asked before. So I think having an inquisitive nature is what drove me to science. And of course, you can do this in any field. You can do this in finance, you can do this, but I think the unique aspect of lab medicine and pathobiology is that you can do all these things. You can experiment, you can have a passion for your job, you can have people around you with the same interests, but I think the unique aspect of lab medicine is the ability to do these while also working towards giving back to individuals in our society that may be disadvantaged by poor health or poor health luck, as is in many cases. So my career trajectory, I think is pretty linear in the sense that traditional, I don't know what you want to call it. I did an undergraduate in health sciences at McMaster. I did a combined MD PhD program at the University of Toronto. And then I did a residency program in LMP with neuropathology. And it really wasn't until that final week before residency applications were due where I kind of made that pivot to really focus on neuropathology. And I think the key thing that I realized, the important things I can look back is that there are very few, even though it's a long journey, it's about a 16-20 year career journey. There are very few, you can count on your finger sometimes, at least for me, certain decisions that kind of got me to where I am today. And one of them was, again, this. I thought I wanted to do anatomical pathology, but when I did my first rotation in neuropathology at Sick Kids and at Sunnybrook with Julia Keith, Cynthia Hawkins, Holiday, I really realized that, I really liked pathology, but I loved neuropathology. And that's when I realized that that had to be the trajectory I went on. And I guess just echoing some of the things that Rita said, I was told if I went into neuropathology, there was no chance I'd be hired. There was no jobs. It's a very subspecialized field. But I took that risk and it paid off, because now I'm doing something that that gives a fire to my spirit every day. Of course, I think that based on this career panel, I had to pick between being a clinician and a clinical scientist, and that was something important. Again, that passion to discover. Again, kind of a risky proposition in terms of job prospects, but something that we can talk about more of how it worked out. The key thing I think that I kind of tell my lab members every day is the asymmetries in your career path. I thought when I was going through that every decision I make is important, and that they're kind of linear things as you go through your career. But the key thing I want to stress is this idea of Price's Law and black swans, where if you kind of look back, you start to notice that there are very few decisions that account for the majority of your success and failures, and it's the key thing is being able to identify them. And in the kind of modern literature, popular culture, these things are called black swans. They're kind of rare events that have dramatic impacts in day-to-day life and how our society runs, and also in our careers. And I can say there's specific mentors that took me under their wings and opened up opportunities. There's specific projects that I worked on that really opened up doors, such as becoming an early adopter of computational pathology. That has been perhaps the most important research-based decision I've made. Discussions and networking with people at kind of LMP research days and all those things that you're involved in. You could have one discussion and it can dramatically change your career trajectory. So it's such an important skill to be able to identify these kind of chance occurrences that change the trajectory of your life. So what do I enjoy? I enjoy mentoring. That's probably if you ask me what gets me to work and what gets me excited, I enjoy working with my team, as I'll highlight soon. This question of how do you balance it? It's really difficult. There's no, this is kind of what my day looks like, or week looks like, constantly. You feel like you've finally found a balance, and someone adds another ball, and you kind of have to adjust to make things work. It's very, very difficult. And I haven't still found the solution, but I guess that's something to consider. So yeah, my career highlights, I love the people I work with. It's such an incredible job. I think I love being a student, but I also love being on the other side and getting to work with trainees, getting to mentor them and help guide them in their kind of career trajectories. Especially since I've been there not too long ago. This is the most rewarding part of my job. That's the biggest challenge, I think, is the mentality from the undergraduate or even professional school. This is the mentality. It's almost like a circus, right? We're these circus performers, and we're asked to do some very challenging tasks, but the success rate that we're expected to do these things is near 99%, right? You need very high marks. You get everything right, even as a clinician. The room for error is very little. And if you have that mentality going to become a clinician scientist, you're kind of open to a rude awakening. And I've heard multiple people, mentors, make the analogy that science is like baseball. Success in science is, if you're batting 30%, you're a superstar. You're not just successful, you're a superstar. And that's been the biggest challenge, is that you have to quickly get acclimatized to getting rejected, whether it be from grants, as we heard from Rita, who's so successful but still had to go through those challenges. And once I went, the second year as a scientist, I got rejected 26 times in 1 year. So those things can be very, very painful. And I think understanding that this is the normal trajectory for a scientist is so important, because you can never, unless you're, TAC-MAC, you're not going to be getting 50% of your papers or grants accepted quite easily. So my advice overall, develop a strong support network, surround yourself with people that have a similar interest and then will support you. Work extremely hard. There's really no substitute. Always try to add value. Whenever you add value in any context, people open doors for you. So it's such an important, and I've seen the same with my trainees. There's when people add value, good things happen. Obviously, no matter how successful you are, always be humble and kind. Kind gestures have the ability to get things back in dividends. Give back, always pay it forward. Help people. Good karma is essential for success. And find your own path. Like my path is unique to me. It's not something I suggest you follow. And I think that's the key message here, is that everyone is unique, and there's no right path. You have to find your own black swan that's going to give you that 5-to-1 return. And that's really the advice I give. We have some more kind of mentorship lectures, a little shameful plug on our YouTube channel. I encourage you to check it out, where we talk about black swans and their impact on careers. And I'll stop there. Thanks for listening. I'm going to hand it over to Susan, and I look forward to your questions during the panel.
Okay, so it's hard to follow Rita and Phidias. They're very inspirational clinician scientists. My path, I guess, is not quite as direct. I am a clinician scientist, and I have now been a clinician scientist for many years. But I have to say, this wasn't where I thought I was going to be. I'm currently a breast pathologist. I've been on staff at UHN, University Health Network in Toronto, for more than 20 years. I have a research lab in the Princess Margaret Cancer Centre Research Institute, where my main research focus is translational breast cancer research. So I look at, I look at breast pathology down the microscope, and that makes me think of questions. I'm very interested currently in how much variability there is within breast cancers. Some other tumors at other sites are very clonal. All the cells look very similar at a genetic level, but in breast cancer, the cells are different, one cell to another. And that, because it's such a frequent phenomenon, that must be important for how the tumor progresses, is a sort of question I've been thinking about and working on recently. I'm also interested in circulating tumor cells, because they are likely the cells that lead to metastasis, which is how death occurs in breast cancer. So I think it's very important that we start to understand those cells and look at alterations that we could block. I mean, if we could stop these cells spreading into the bloodstream and block breast cancer so it stays in the breast, it could be quite disfiguring, but it wouldn't kill the patient. And so that would be important. And also I'm interested in this transition from breast cancer when it's contained within the ducts, what we call pre-invasive breast cancer or ductal carcinoma in situ, and what triggers that to invade, to go beyond the wall of the duct. And these are all questions that come up, looking down a microscope, looking at patient samples, and that's how most of my research questions arise. I also value the multidisciplinary teams that we work in, and in breast cancer, I work very closely with medical oncologists, surgical oncologists, radiation oncologists, breast imaging specialists, radiologists, and geneticists. And this multidisciplinary approach is very stimulating, and it leads to a lot of questions, particularly around imaging specimens. So imaging intraoperatively or improved radiological imaging in terms of screening, or using radioisotopes to label tumor cells within patients, which can be used to target tumors. So that's my research, and I'm actually not going to spend a whole lot of time talking about my research because I thought I'd focus my talk on how I became a clinician scientist. So if anyone's interested in my research, they can take that offline and we can talk about it some other time or later. So this outline of what I'm going to talk about is how I got here. I'm going to give you my career path, which is obviously slightly is different from Rita and Phidias's, but everybody's different. I'm going to talk about the life of a clinician scientist, the pros and the cons. While it's a fantastic career and I highly recommend it, it's not without difficulties and problems. I'm going to give you some advice as to things to think about if you would like to become a clinician scientist, and I'm going to finish up with a short summary. So setting out, as you can tell from my accent, I grew up in England. I grew up in a medical household in a middle-sized town in northern England, a grey town filled with derelict mills. It was a quite depressed place to grow up. But I grew up in a medical family. As I say, my father was a general surgeon in a large community hospital, and my mother was a nurse. She was actually an immigrant. She's come, and it was always just assumed that I would go into medicine. And I think this is an issue that's people talk about more these days, that people want more exposure to different careers and to explore different things. Not that I have any regrets about going into medicine, but there was never any suggestion I would do anything else. And I had no exposure at all to research in my high school time. Anyway, following high school, I got into Cambridge, which was a fantastic experience and really changed my life, opened my horizons. I met people doing subjects I'd never heard of, and I did get my first exposure to research at this point. I give a quick quote this is a quote from Richard Branston. The original quote was actually business opportunity buses. There's always another one coming, but I thought I could trim off the business and keep opportunities are like buses. There's always another one coming. And I think this is a theme that's resounding with what Rita and Phidias said, that, we have to be aware of these opportunities that come up intermittently during our lives and be ready to take them or not, but at least to recognize them and give duration. And again, going back to these turning points that Phidias was referring to, this is my career trajectory. So did have the years, but I didn't want to myself. So the time that I left high school, I was 18 in England, and I got into Cambridge. Was medical school at Oxford and Cambridge were the hardest programs to get into out of high school. So that was quite an achievement. But as soon as I hit Cambridge, I realized that there were many people much brighter than I was, and so that was kind of a bit of a setback initially, but it went well, and I had a great time. Obtained my medical qualification. I stayed in Cambridge for my full medical training, which a lot of people moved to London halfway through, but I was quite sport, and I was competing for my university at orienteering. I wanted to stay for all 5.5 years in Cambridge and did my clinical school at Addenbrooke's. Because my father had been a surgeon, and that was really the only thing I had any experience of, I wanted to be a surgeon, not surprisingly. And I got house jobs in Norwich and Cambridge, which was good and putting me on the right track, because there are many house jobs in Cambridge. It's not a huge teaching hospital. It's really just a district general, because Cambridge isn't a huge place. But during the time I spent in my first year, I realized that surgery wasn't for me. The career prospects were poor, and certainly as a woman, there would be no opportunity to have a family if I wanted to be successful at that time. So then I sort of sat back and thought, well, what do I want to do? I was a physician, but I really didn't know what I want to specialize in. So then I became a senior house officer, which is junior resident in emergency medicine. And in England, we called it accident and emergency. And I went and did that for 6 months in London. And that gave me exposure, real life exposure to many different specialties. And really from that, I gained a lot of experience on what I didn't want to do. I didn't want to do pediatrics because really you would deal with the parents all the time. And it was a lot of the problems were with the parents. So many things I didn't want to do, but I realized that I was very interested in the diseases that people came in with. You know, being in London, people would come in off the planes at Heathrow, you know, dengue and sorts of really actual conditions, and I was making diagnoses in the ER, and I would follow up to see if I was right and what had happened to a lot of patients. I hadn't really considered pathology at all. At Cambridge, the pathology department were very intimidating. They were very bright. They were very hard on us as med students. And it never occurred to me I could be a pathologist. So it was only that was the first time when I did emergency medicine that I thought, well, maybe I could do pathology. And so I followed time in emergency with a year as an SHO, senior house officer, in general pathology on the outskirts of Manchester. So that was an important, put a star there, that was an important time for me. And yes, I did like pathology, actually. It was really interesting and I could see myself doing it and it would be a rewarding career. Going back to the time in Cambridge, the staff, kind of the turning point in my life there was, I met my husband at medical school, who was a Canadian who'd done an undergrad in Calgary, where he came from. And he wasn't sure what he wanted to do. He thought, but a family friend had gone to Cambridge and he thought that might be interesting. So he applied and got in, and he was studying medicine there, and that's how we met. And so following the year as an SHO, we got married, and he wanted to come back to Calgary, his family there, and we thought we'd give it a try. And I got into the residency program in anatomic pathology in Calgary and did a rotating internship prior to that, and had a fantastic 5 years training in pathology in Calgary. It was a fantastic program, quite a smaller program than the Toronto program, very, a lot of social activities around the program, now to the country as well. So I appreciated being sort of welcomed into a community. And so, as I say, I had a fantastic time. However, at the end of that, my husband wanted to specialize. My husband did internal medicine and wanted to specialize in allergy and clinical immunology, which they didn't have in Calgary. And he got into the training program in Toronto, a 2-year program. During my residency, I realized that, you see different career laid out ahead of you, and you see people above ahead of you, what types of lifestyles they had. And I really was kind of gravitating towards an academic career. I wanted to be at a university. I was interested in research. I got involved in some research projects during my residency. And I got a 2-year residency in Calgary. They were keen for me to stay. I was going to do a 2-year research project funded by the Alberta Heritage Foundation at the end of my residency. But as time ticked on, and my husband was going to be in Toronto for 2 years, and I was going to be in Calgary, it just seemed, it was going to be so hard, and we hadn't have enough money to fly to see each other very often. And I was finding out one day toward the end of my final year in residency with actually a GI pathologist who knew somebody in Toronto who just got a grant funded, and they needed a fellow to be involved in the project. And they thought that might be something that might be interesting. And it was one of those right place, right time. And I leapt on it and said, yes, that sounds fantastic. And he contacted the person he knew, who was a GI pathologist in Toronto, Mark Redston at Mount Sinai, and the project with Irene Androulis, also at Mount Sinai, a basic science researcher working on breast cancer. And they offered me the job, and I came out for a 2-year molecular pathology fellowship at Mount Sinai. And that took me into the next phase of my life. And the other thing during residency, as I say, I'd become interested, as I didn't have a particular interest in breast cancer going into my residency, But as a resident rotating through all the different areas that you go through, I gravitated towards breast pathology. I found that really fascinating, but also renal pathology. So coming out of the residency, if an opportunity had come up in renal pathology, I may have gone in a different direction. But anyway, regardless, I started my 2-year molecular pathology fellowship at Mount Sinai. And I really got into the research. I was told initially, well, you're here for 2 years, why don't you enroll in the graduate program and do an MSc? And I thought, well, sure, that sounds great, why not? And I started in the MSc. I think Dr. Candel was actually on my supervisory committee. She's a very inspirational figure, I remember at that time, and as she still is. And during my master's, I realized I wanted to go further with research. And so I reclassified into the PhD program. I needed additional funding. Sinai would fund the first 2 years that they agreed to, but beyond that, I needed additional funding. And so I applied to the National Cancer Institute of Canada and got an MD fellowship from them to fund the further 3 years of my training. And at the end of as I was coming to the end of my training, they were looking for a clinician scientist to work on breast cancer at UHN. So it was one of those sort of right place, right time things again. But again, I think just being open to opportunities and being on the lookout for them. And so that's how I ended up on staff at UHN. And more than 20 years later, I'm still here. So that's my story. Talking about life as a clinician scientist, it's a fantastic job. It's a fantastic life. It's so fun to be able to ask questions in your work as a pathologist, and then go to the lab and answer them. And that's tremendously exciting. It's tremendously fulfilling. It's something that not many people have that opportunity to go that whole circle of inquiry. You're able to influence the direction research takes. I mean, nobody tells me what grant I'm going to write. I'm able to say that I think circulating tumor cells are important. I'm working on a grant at the moment that I'm going to submit in the next couple of weeks on intratumor heterogeneity and triple negative breast cancer, which I think is hugely important. And by working on that, I can tell other people about it, get other people to get involved in it. So, that's very satisfying to have that degree of influence, because some of the time you're working in research, as I'm sure a lot of people in the meeting know, is more flexible in terms of scheduling. It's nice to have that control. It's very rewarding interacting with scientists and students, discussing new ideas, challenging existing ideas. It's so satisfying, and it really helps you grow all the time. It's fantastic being involved in a dynamic, evolving field, as areas of research are. And then, there's the joy and satisfaction of success when you get that grant or when you get that publication. That's a fantastic feeling. So there are a lot of pros to being a clinician scientist. However, it's not all good all the time. There is a constant pressure to obtain funding. I've been lucky. I've had funding from granting agencies, charitable agencies, but it can get very anxiety provoking. I've stayed awake late at night sometimes, worrying about where the next grant's going to come from. As Dr. Candel says, as we said, sometimes you can go to other groups, but that very much depends on the type of research you're doing. The research, the sort of basic biology of breast cancer, it's hard to get funding from other agencies. If I was a bit close to the clinic, I could go maybe to drug. That's not something I've explored, but I guess it's a possibility. It's very difficult to get and because as a pathologist, you're paid as a pathologist, and many hospitals see that as something that if they're paying you to be a pathologist, they want you to be a pathologist full time. As we've talked about, you have to train as both a physician and then a specialist in pathology or another specialty, and also train in research. And so you end up starting your career later than a lot of your colleagues, which in the early days is not such a problem because, they're knuckling down to a sort of, 9 to 6, 9 to 7 work day. And you're maybe, I was engaged in research. I was doing my PhD, which was a tremendously fun time and really stimulating. And I had a lot of control over what I did. But now I ended up starting on staff later. And then you think about, saving for pensions and things, and you've got, less years. So that's something to think about. And it can be difficult to integrate this sort of prolonged period of training and also the demands of being a clinician and a scientist to having a family. And I think that's particularly hard for women. And then you have to compete with others doing both roles full time. I mean, I can't be any less a pathologist than my colleagues in breast pathologist. I have to be as good, if not better, than they are at what I do clinically. And also equally, you have to compete with scientists who are doing science full time. So, there's no, there's no allowances for what you're doing. So given, if you still want to be a clinician scientist, how would you go about it? So, the first thing is you need a passion for research. You've got to make sure it's something you enjoy and you have a talent for. I mean, you need both things. If you want principal investigator, I think you need to spend a dedicated period of time engaged in research full time. And the challenge is when to do this. Do you do it before, if you do it too early, if you do it, say, before you go to med school, if you did a PhD and then went to med school, the difficulty is that your research, you would be kind of out of, what you've done in research would be kind of out of date by that stage. The way I did it, where I did my PhD after my residency, worked well for me. But I mean, it may be hard to come back into a clinical job when you've spent 5 years away. It depends on the job and what they're looking for at the time. If you want to become a clinician scientist, it's very important to negotiate a startup package that includes protected time for research, lab space, and funds to get started. You likely won't get a grant immediately. You need preliminary data and to get that first grant. So you do need some money to get started on your experiments, and you need probably at least a full-time technician to help you to do the experiments you need to get started. So that's important. And if you don't start with protected time, it's almost impossible to get it afterwards. It's very hard. So if something you want to do, I would encourage you to demand the protected time up front. You need to become good, very good, I'd say, at writing grants quickly. So if something you're thinking about doing, look out for grant workshops. I went to a very good one. I'm not a good writer, as you could see from my slides. I think I spelt father wrong. I was a bit of a borderline dyslexic, I think. I went to a very good grant writing workshop through the AACR, the American Association for Cancer Research. And I took that as I was close to finishing my PhD, and I found I'm not a naturally good writer, and I found it very helpful and really helped elevate my grant writing skills. A mentor can be very helpful. It's difficult to succeed without one, so look out for, as you go through training, look for mentors that can help you give you advice in your career path. And then look for agencies that provide support. As I say, my PhD was funded in large part by the National Cancer Institute of Canada, which doesn't exist anymore, but CIHR does have a clinician scientist program, and I think that has 2 phases even to take you through different phases of being a clinician scientist, but certainly look out for agencies that will support you. So that's basically what I had to say. In summary, go to my summary, sorry. So I guess life is a great adventure. I haven't ended up where I thought when I was in high school and even at university. I would never imagined I would end up here. although I'm very happy that I have. So you may not end up where you expected, and that may not be a bad thing. Seize, or at least seriously consider every opportunity that comes your way. The life of a clinician scientist is hugely rewarding, advancing patient care and scientific knowledge, as well as being a member of the research community and training junior investigators. They're all very fun and very rewarding things to do. So with that, I've finished. I'll say thank you, and I'm happy to take any questions.
Hi, everyone. So first of all, I'd like to thank everyone and all the speakers for such amazing talk. As a student myself, I found them extremely informative. So we'll get started on the Q&A right away. These questions include the ones that were submitted by the attendees during the registration and the ones that were asked during the chat. And on that note, if you have any questions, please don't hesitate to drop them in the chat. So our first question to our speakers today is, why did you choose to combine an MD with research? So for that first question, I will ask Dr. Candle to please answer it.
Well, I must admit, I don't feel like I had a choice. It's an interesting question. When you are a physician, there are so many questions that rise to the forefront that you want to help answer. And the combination of doing research and medicine seemed like an inevitable thing to do if you were going to answer those questions in a way that would contribute to knowledge. And so for me, it was important to combine both. I wouldn't have been satisfied with one or just the other.
All right, thank you, Rita. Would Susan and Phidias like to add on to their experiences to the question of why they chose to combine an MD with research?
Yeah, I mean, as I said when I was speaking, I never set out to do research. I was going to be a physician, and it was only during my residency that I realized, that I learned about research, and that I realized how rewarding and how exciting it was. So I started as an MD, but decided that, just being a clinician kind of wasn't enough, really. I think that's maybe how I got here.
All right, thank you, Susan. We'll move on to our second question. So, how do you keep up with advances in clinical practice and advances in research, whether it is new technology or whether it is new knowledge? So Phidias, would you like to answer that question?
Yeah, so that's probably the hardest part of being a clinician scientist, because as Susan said, there's no, you're competing, or I don't want to say competing, but you're held to the same standard as your clinical colleagues and as your research colleagues. So it's just kind of mission impossible about trying to, do two jobs at once, basically. And I think what has worked for me is, there's one advantage, is that most new advances in clinical practice is research. And so when you do research and you talk to people at meetings or you attend conferences, you see what's in the pipeline. A lot of molecular medicine is what people are doing research on. So in a way, your research gives you kind of an early look into what's going to become standard clinical practice. The other thing too, I think that's important, that has worked for me well, is you start using your situation as an advantage. So all my research questions, or the majority of them, have a histology focus. And so that helps when I create a cohort for a research study, I'm looking at slides, so I'm maintaining my clinical skills, and I'm also differentiating myself from my research colleagues. So I'm never directly competing with them on a grant panel. So I think that's the key, at least for me, for survival, is finding the kind of interface where this disadvantage becomes an advantage.
All right, thank you, Phidias. Would Rita and Susan like to add on to how you can keep up with the clinical advances in research?
So I'm just going to add to Phidias, because everything he's spoken is the truth. But you know what? For me, I can't help but read and keep up. I always want to know what's going on in my fields. I want to learn new things that I might apply to what I'm doing, I'm studying, and advance that research program. So my problem is not reading and not keeping up, and spending time doing other things, I suppose. But for me, it's quite the opposite. My joy in part comes from sitting down and reading both clinical and research topics.
Yeah, I echo what they, but I think also for science in particular, it's really important to network and to be part of the scientific community. I mean, we're lucky here in Toronto to have so many science institutes so close together. Hopefully when things start up again, we can have more in-person interactions, because I think that's sort of scientific discovery and hearing people talk and challenging other people's ideas. I think that's very important for science. And another thing is, once you become known in an area, you get asked to review grants and review manuscripts, and that also helps you see what people are working on and what ideas are sort of coming forward. So there are a lot of ways of keeping up. Yeah.
So I guess the key takeaway from that question, from what I have understood, is reading and networking. So with that, we'll move on to our next question, which is, what is the best career advice you have ever been given? So Rita, would you like to go first?
That's a great question as well. I would say the best advice I gave was focus. You really need to focus. One of the things that we do is we get interested in an area, and it just expands and grows exponentially if you let it. And the key is to keep focus, so that you keep your research focused, and you actually then have the ability to keep on top of it all. So my greatest advice that I ever received was learn to focus.
Thank you, Rita. Would Susan and Phidias like to include any advice that they were given?
I've never had a whole lot of very useful advice, to be honest. I guess the best advice I've had is, find a job you enjoy and you'll never work a day. I think that's true. You've really got to enjoy what you do, because you're putting a lot of hours in as a clinician scientist.
Thank you, Susan.
Yes.
You have advice?
I think I agree. The single most important advice is you have to be able to go into a room, and you have to be so passionate about what you're doing that you need to tell people why what you're interested in is so important and so interesting. And I think if you don't have that passion, it's just so hard to do this job. I'll give another piece of advice, just to kind of not give the same one that Susan gave. Is the most important thing that I did is learn how to write well. So much of our job is it's good to like what you do, but this academia is peer based, and it's not just the person with the best idea that succeeds. It's the person that can communicate that idea in an engaging way to others. And when I, the biggest difference when I got those 20 straight rejections, the thing that I realized is I need this person reading it to be as excited about what I'm doing as I am. And if you can do that, if you can learn how to convey what you're doing in an interesting way, I think it's easier to succeed, because people like passion. And if you can communicate passion, it's easier to excel.
All right, thank you, Phidias. With that, we will move on to our next question, which is, how did you deal with setbacks in your career path, and how has that impacted you? Susan, would you like to go first?
Yeah. I guess I didn't, because as I said, I didn't really have a specific path. So, say I've had a setback, I kind of, jumped from an opportunity to another, really. I came to Canada. I wasn't sure if it was going to work out here. I would have gone back to the UK had it worked out here. I went to Toronto to do the research fellowship for 2 years, planning at the time to come back to Calgary, but things, I really got into the research and ended up doing a PhD and stayed for. I guess, I've had a lot of grants being funded, but and I never really had a huge setback, I guess. I think the hardest thing is getting grants rejected, I guess. It's very hard to read those reviewers' comments when they come back. Often I have to put them away and bring them out, a few days later when I've got used to the idea. So, yeah.
Well, I think that's great to not have any setbacks, Susan. So I think you're good. I know Rita mentioned in her talk that she, as a woman in STEM, she'd had to deal with a lot of setbacks. So Rita, would you like to expand on how you sort of dealt with them?
So I think that setbacks are a result of living. And if you're living a full life, you're going to experience setbacks. But they're setbacks. The only thing that really truly matters, I think, is the people you care about and your family. And if they're all right, those setbacks are just setbacks, and you learn from them, you grow, and you move on.
Thank you, Rita. Phidias, would you like to include your experience as well?
Yeah, I agree. I agree with both Susan and Rita. Like, it just, there's a good quote, if you're not getting failures, it means that the goals that you're setting are not high enough, right? If you're 17 and you're playing under 18 tennis, for example, and you have a 50-0 record, it means that you're not playing at the level that you should be. You should be playing, amateur or pro, right? So I think failure is a metric that lets you know that I'm doing something difficult enough that the reward is enjoyable. But failure is tough. I'm worse than Susan. I don't even read the reviews from. I'm too scared to know what my peers think is wrong with the grants they reject. But I think the important thing to realize is it's a very subjective field. Like, although science is supposedly objective, we're just not very good. I think that's the black swan mentality, is that we're not very good at understanding what a transformative idea looks like. When you hear all the people who win Nobel Prizes, that specific paper, the landmark paper, got rejected by top journals. Even some intermediate journals rejected it, right? So I think it's important to realize a rejection in most cases is just a subjective opinion about your passion. And sometimes I'll submit the same grant to another agency and I'll get triaged in one and I'll get ranked. So I think the key is just don't take it too personally if you can, because it's an imperfect science. Some of these metrics that we use for success.
Thank you, Phidias. I'd actually like to go back to Rita again. So in your presentation, Rita, you told all of us that you face many barriers because you're a woman. So how does that inform your leadership style now?
Well, I'd like to believe that it is made me sensitive to all bias in all situations, and make sure that I step up and speak out to ensure that there is no bias. And I think that's the way I've responded to it.
I think that's a great way. Now we're gonna address some questions that were received in the chat. So Hannah asked a question about how necessary do you think graduate studies, so both MSc as well as PhD, are today for doing research as a clinician scientist? And do you think there are still significant barriers if you don't have either a PhD or MSc to be a clinician scientist? So Phidias, would you like to take a lead on that question?
You know, I think it's I think a PhD is a powerful degree, as is a master's. But it's not necessarily the paper that you get; it's the experience that you get during that time. And we talk about it with my lab a lot. What happens if I don't go into academia? How should I market myself? And I see graduate studies. You learn how to make very important decisions, and same with medicine. Is this person have cancer, or is it a benign condition? Should I spend my supervisor's grant money on this experiment, or that? Some of these experiments that we do cost tens of thousands of dollars, and we leave it up to the trainees to decide what to do. So I think that's the key skill is, life is about making tough decisions. And I think you can learn that in medical school, you can learn that in PhD training. And of course, if you do both, or if you do one longer, you have more of that kind of maturity to make those challenging decisions. Can you learn those in other ways? I think so. I mean, I think, I don't think there's a specific recipe, but I do think there's some, those are the key skills that I think you learn in graduate studies.
Thank you, Phidias. Would Rita and Susan like to expand on that, whether like a graduate study, PhD, MSc is necessary to succeed as a clinician scientist?
I'll take it. I think it is. I don't think you'll get a clinician scientist without it. I know there are some people with masters around that are very successful clinician scientists, but I think you just make it harder. And as a woman, I think it's even harder. So I think as a woman, you need to have a PhD if you want to have lab space, which is a big distinction. Rita, what do you think? Sorry.
Yeah, I mean, I encourage people to go on to do advanced degrees. I think research is a wonderful experience, and it really trains you, as Phidias said, in critical thinking and logical thought. And those are skills that will carry you through whatever field you go into. And if you choose to do science, I think, obviously I didn't get a PhD, and in the end, I don't think it held me back. But I think in this day and age, if you want to get that training, it's much easier to get support to do the training if you go through the PhD program. It's harder to find money to do it otherwise.
And when you look at grant review panels, it's predominantly scientists, because a lot of clinician scientists can't get away to be on panels. So there are more basic scientists in grant reviews, and they end a career path of a basic scientist better than a clinician scientist often. That can be
Thank you. We've got about 3 minutes left, so we're going to quickly zoom through the questions that we have now. So next question is, are there any tips to balance the progress between clinical training and research early during career development? So would any of you like to answer that question?
There's no tips. You have to commit and go through the training of each area, as an MD, as a researcher, and you need to do it. So the only advice I would give is to enjoy it. It's a great time of your life.
Thank you, Rita. And then the next question we have is, did any of the panelists ever consider an alternate career, such as industry? And what made them confident that their decision to pursue being a clinician scientist? So does have any of you ever considered like an alternate career in industry?
No. No, I have never considered it. As a matter of fact, I've turned down such offers. I would never, for me, having the freedom to pursue whatever it was that I was interested in was very important. So industry was not a place for me.
I'll say one thing. I do promote it within my trainees because I think it's very tough. There's not a lot of academic jobs. So I thought about it. I don't have any experience in industry, but we see a lot of the kind of transformative companies in today's era. Being smaller startups, independent of universities. So. Even though, maybe we don't have experience and it may be something we didn't pursue, I wouldn't, I encourage people to at least explore it through things like MyTax. There's, there's internships and fellowships that you can get funding to at least try it because, we're somewhat biased, right? We're in academia and, we're probably here because we've somewhat excelled in academia. So, it's important to, I think, to experience it because maybe other people know something that we don't.
That's a good point, Phidias. I think that's a very good point. Okay.
So thank you everyone for answering all of those questions. We are reaching the time, so it's 6:40 and we don't want to go over time. So I see that we have a few more questions in the chat. I'm sure if you'd like to reach out to our panelists, they would be happy to answer these questions. And I will hand it back to Gino.
Maryam, thank you so much for moderating the Q&A session. It's often the most difficult part of a talk and you've done it like a professional. I'm really happy with how that's gone. The questions were fantastic. Thank you to our audience for providing them. And thank you so much to our three panelists, Rita, Phidias, and Susan, for sharing your journey through the choppy and never boring waters of medicine and science. I was incredibly inspired, and the take-home message, there's many, but one, take those opportunities when they arise. Please, to everybody on the call, fill out the evaluation form. It'll be either in the chat and it'll be emailed to you at a later time. And look out for our next alumni panel sometime in the late spring in May. We'll be bringing together another 3 wonderful speakers to talk about some alternative paths in a career after LMP. Many thanks everyone and enjoy the rest of your evening.